have anything to say regarding the subject or any opinion, till the point it is a scientific opinion, even if it's a personal informed opinion, you have to discuss that with me, and do not hesitate in doing that, because my classes are going to be discussive. They will not be a monologue, and I will appreciate if you all will help me in that. So I can expect a good participation from all of you to learn the subject. Say yes or no.
Yes,
Yes, sir.
Yes, sir.
This is like an oath I am signing you all for. We keep it. Anyone who has not spoken for introduction, I think everyone has introduced, right? And I have introduced to all of you. Okay, so let's begin. See, the way I'm going to teach you biology comes from both my student teaching and research experience, okay? And I have realized one problem that almost every student, whether he or she likes biology or do not like biology, faces regardless, is that biology, you have to learn a lot, right? Am I correct? There are many facts associated with biology that you have to learn, and sometimes it becomes overwhelming and difficult, correct? Especially for entrances, right? Yes or no?
Yes, sir.
Yes, sir.
So my sole purpose will be to teach you biology in a way where your reliance on memorizing the facts decreases, and a concept map of the subject gets in your mind, just like you remember a movie or a song. You never have to mug up, sit, take the lyrics or take the story or the screenplay of the movie and remember it, correct? You just see it, there's a concept map in your head that this happened, this person or he or she was the protagonist, this is the story, these were the songs, and it's just in your head. We'll try to put biology like that. Okay, so the first chapter in your book though is reproduction, but before reaching there, I'll be spending some time on some basic foundational concepts of biology, okay, that you have studied in class 9, 10th, 11th. And this, all the introductory classes are an opportunity for you and me both to know each other, for me to understand where you lie, all of you as students. And I'm not going to just teach you restricted 12th class syllabus, okay? Because in a longer run, I will be preparing you for anything and everything that you have to do in biology, for entrances, your school examinations, or to become a researcher or anything, okay? So feel free to discuss, feel free to ask questions, even if it is from 11th class or somewhere else you have studied about biology, no problem, okay? So before we begin reproduction, I would like to go a little bit back to the chapter in 11th class that you studied, that's called life processes. You remember this chapter, everyone?
Yes.
Life process?
Yes, sir.
Yes, sir.
Can anyone just define for me what does this word means, life process? Yes, anyone, whoever starts speaking can speak. I'll not pick students right now in the first class. So what's life process? Let's just hear from you. Anyone? If you'll not speak, then I'll pick.
So does it mean the functioning of life?
Yes, Samson.
It means the functioning of life.
Processes of
Sir.
life. Yeah, that's like defining.
Life process.
Yes, Shin.
Life process is in which like it is necessary to maintain life in organisms.
Yes. So you used a very good word, maintenance, right? So life processes are those processes which are required for maintenance of life of an individual, of an organism, right? You use the word organism also, that's quite good. Okay, so in my class, I forgot to tell you this one thing that I always tell in the beginning. See, why I was talking about difficult things to remember in biology was because of the words that are there in biology. Many technical words which are there in biology are not derived from the English language. That's why they are difficult to remember at times, including your scientific names of the organisms, correct? In 11th class, you must have read about that, the binomial nomenclature. But binomial nomenclature is not the only difficult thing in biology. Apart from that, we also have difficult terminologies, technical terms that are derived from English. But if you understand those terms, see, in biology, there are two things, okay? So till now, whatever kind of biology learning you were doing, I would like you to be open to unlearn it if it is required. So the most important thing in learning is to be able to unlearn, because this is how our syllabus is designed. This is how we teach students right from the beginning. So in primary classes, we teach students something that looks like a fact, but when they go to higher classes, we tell them that, see, this what we taught was not exactly correct, and this is what is exactly correct. And this is how science have developed. In chemistry, you all must have read about how the structure of atom was discovered, correct? Someone thought it's like a pudding, someone thought it's like a watermelon, plum pudding, then someone said there will be orbits, discrete orbits, and at the end we reached to the model that we follow today, correct? And that's true for everything. That's true for biology, chemistry, physics, even mathematics, and all sciences. So you have to be open to unlearn. I'll tell you where you have to unlearn what you have learned before. So first thing that you have to ask in biology is what? What are we studying right now? What are we learning? And then how? How it happens. So science is mostly about what and how. It's not about why most of the times, because why do not have a concrete answer in biology. You know, then we go into the philosophy of science, and philosophy of science is very, it's not anything concrete. It's very subjective, person to person, and whoever has more experience in science have a different philosophy of science. So the part what can get to some extent clear if you understand the words, biological words, or I call it terminologies. I have not invented it, I just use this word. And the part how is the part that will fetch you marks both in entrances and in your examinations. The bulk marks comes from how. How is the process of anything that we pick up. For example, if we pick up heart, then how does heart function? How is it developed? And everything associated with that. But this terminology, what you have to do is you have to maintain a copy for my classes, a separate copy, and leave the first 3 to 4 pages of that copy blank. Okay. And you have to make a division in the middle. On one side, you will write terminologies, on the other side, you will write meaning. So what basically I want you to do is, by the end of this year, as we will progress in biology, you will have your own mini dictionary of biological terms that are going to be helping you in all entrances and exams. Is it clear, everyone? Say yes. If it's
Yes,
clear.
sir.
Yes, sir.
All have to maintain this terminology section. This is how we learn biology, where we, because these terms will be used multiple times, and something that repeatedly we hear, it's basic neuroscience and the science of memory formation. So from neurobiology, I can tell you, we remember songs very easily because of two things. One thing is the repetition, the other thing is the beat. So anything which is in flow is easy to remember. So if we start understanding the process of biology in flow, like by in flow, I don't mean that I'm going to rap for you here, or I'm going to sing for you here. I'll do it if it's required, don't worry. But what I mean to say is we'll talk about those concepts in a coherent manner. So if we are studying one aspect of biology, all other aspects are some or the other way related. So at many a times, I'll take you out and ask you, remember we did this, and this is how it is connected to this particular thing we are studying. Okay. So by the end of this whole year, you will not be needing to memorize a lot of terms. A lot of terms will just become the part of your basic biological vocabulary. Okay, everyone? Just like me.
Yes, sir.
Yes, sir.
Okay, very good. So this you have to do, the first thing in my class. Second thing I've already told you that you have to be discussing. So we were discussing life process, and someone said that life process are those processes that are important to maintain life of any organism, right? And in that life process, you have studied reproduction as one of the process, correct?
Yes.
Yes, sir.
It's important for the maintenance of life. And when I say maintenance of life, what do I mean? So if,
Survival.
yes, Fatima Munir. So I do not mean survival. You know, this is what I was coming up to. Thanks for introducing this term. So I mean to say that something could be a life process. Is it possible? It's a question to all of you. Is it possible that something can be a life process but yet not vital for survival?
Yes, sir.
Yes. Why?
Yes, sir.
Yes, Fatima. Why do you think so?
Because like reproduction is not vital for survival. You can live without reproducing.
Yes, correct.
Even you can live without.
Yes, correct. And even there are organisms that are incapable of reproducing, like sterile and infertile organisms, but they do live, right? So life processes are the processes that helps to maintain life, but are not necessary for the survival of the organism. Okay. But still, what is okay? Samson is asking a question. Samson, you can ask the question here also. Okay, if you can speak, that will be great. It will save some time. So you're asking what I was saying about making the book by the end of session. Oh, I was saying that if you will make this terminology section in your copy, the first few pages, by the end of the session, like this year, you will be having a mini dictionary of your own ready and in your mind, okay, along with on those pages. That will make biology very easy for you and you have to remember less. Okay, Samson?
Okay, sir.
Yeah. Okay, let's come back to reproduction. So I said that you said that it's not important. Fatima said it's not important for survival of organisms. Then what is it important for? Why are we studying about reproduction then?
Sir, so that the species do not get extinct.
Yes, correct. It's about the survival of or the continuation of species, not about the survival of organism. Okay. So reproduction is important in a long run, in a population dynamics. It's not important that much for an organism's point of view. In fact, reproduction is a very, very energy consuming process. And oftentimes when it comes between survival and reproduction, organisms have to choose one. They can go for survival with by stopping reproduction. You know, bacteria do that and a lot of other organisms do that when the conditions are not favorable. Correct? Opposite
Yes,
of
sir.
this behavior has also been noticed, and I'll talk about that also in biology. You know, we were talking about plants being very complex organisms. Even many animals often, just before they feel that or the environment is going to be hostile, they utilize all their energy in reproduction and then they die, because they want to ensure that even if I die, I ensure that I make progenies, a lot of progenies if possible, from which at least some can survive. Now, this is not a conscious decision on an organism's front if it is not a human, because every organism does not learn about biology, they just live biology. But still it is happening in biology. This is how it is shaped. Okay. So it's very interesting subject. Every one of you said that, and I completely agree with you on this. And I hope for a good batch and a good discussion session in these classes. Okay, so let's start with defining reproduction. Everyone, please write down. Reproduction is the process. So I'll be dictating certain things and I'll be writing some certain things. Is it okay for all of you? Anyone who wants me to write everything or dictate everything? A mixture will work?
Yes, sir.
Yes, sir.
Write down. Reproduction is the process by which organisms produce offsprings. Organisms produce offsprings which are similar to the parent organism, in their adult form. This term here is important to begin with. So why do you think I use the word adult form?
Because parent organisms produce offspring from adult parent.
No, parents have to be adult, of course. They have to be sexually mature to be able to produce offsprings. But this definition means, can someone read it again, please, for me? Just for me to check if I have. Yes, Fatima, please.
Is the process by which organisms produce offsprings which are similar to the parent organism in their adult form.
Yeah, the word there is for offsprings here. If you are confused, write down the last word there in their adult form. Just before adult, you can write offspring. It is for offspring. So I mean to say that by this process, the offsprings that are produced look similar to the parents in the adult form. So
can you repeat the definition of reproduction again?
Yes, that will be a little difficult for me. Can someone else? So because when I dictate you terms, they are not like right from book or somewhere. Yes, Shin, can you just repeat it?
Reproduction is a process.
Repeat because so that Mehraj can write.
Reproduction is a process by which organisms produce offspring which are similar to the parent organisms in their adult form.
In their adult form. Done, Mehraj?
Thank you, sir.
Yes. You have to thank Shin. Okay. But why did I use the word adult form here? Can anyone guess? Mamana, you can also pitch in, because if there is something we have already discussed, you can pitch in and help your batchmates. It's better than me speaking all the time. See, I use the word adult here because not all organisms' offsprings, like the babies, look like their parents. For humans, a baby of a human looks like a human, an adult human. It's just tinier. It's a tiny human, little human, but the anatomy and the physiology is similar, correct? If not exactly same with respect to bones and certain organs, but still it is morphologically and anatomically similar. So a human baby looks like a human baby. But if you see a frog's baby, a frog's offspring, what do we call a frog's offspring?
Tadpole.
Yes. So frogs' offsprings are called tadpoles. Do they look like frogs?
No, sir.
What do they look like? What do tadpoles look like? No one has seen a tadpole?
No, sir.
Not even in pictures?
Like very
Sir, just in
small.
Mr. Bean.
Anyone has seen a tadpole in real? Who has seen
So
tadpoles?
they are small worm-like creatures.
Worm-like. Okay. So you have not seen a tadpole. Thanks, Mehraj.
It's like black in color, black brownish in color.
Yes. You have seen it or you have?
Yeah, I had frogs before.
Okay. But which other organism it looks like or resembles?
Looks like a sperm.
It looks like sperm. Oh, that's very innovative. Yes, it does look like a very big sperm, which can be seen with the naked eye. That's true. But any other macroscopic organism around us, if you see fish babies with tadpoles, you will not be able to differentiate between the two most of the time. So they do look like small fish in water, right? Or babies of the fish. So frog offsprings look very similar to fish offsprings and fish because they do not have appendages. So a tadpole looks something like this. It has a head, and it has a body, and then it has a fin, right, at the end. Of course, it has, and this look like larvae of the fish, correct, everyone? No
Yes, sir.
limbs,
Yes,
no hand,
sir.
no leg, doesn't look like a frog. So there is this kind of development also in reproduction where the young ones do not look like their parents, but still they are their parents' young ones. Another example could be insects, especially butterflies. You all have seen butterflies, very, very colorful, very beautiful.
Yes, sir.
Yes, sir.
What are the offsprings of butterflies?
Caterpillar.
Caterpillars. Do caterpillar looks like butterflies?
No.
No, sir.
What do they look like? Someone in the last answer used the word worm. Vermin.
Yes, so they look like worms.
Yes. So caterpillars are worm-like, right? They wriggle and they move like worms, like an earthworm or any other worm, right? So
Yes, sir.
sometime they develop into different, going through different stages of development. They finally come out of their cocoon or their pupa form, and they look like a butterfly. So this kind of development where there are stages which do not look like the parent at all, and over time they change, is called indirect development. What it is called? Indirect. So indirect development we will study in detail. I'm just making foundation for you in this class for this whole chapter that we will be continuing with. So indirect development is where organisms, the young ones, go through different stages where they change, they transform from something very different to something very different.
So isn't that called metamorphosis?
Yes, I was coming to this. So indirect development is the term, and we also use terms like metamorphosis. Now this is your first term, which is a very, very complicated term. How to remember this? See, what is meta? You all know about it. Meta. What is meta? You all are just about to enter metaverse. You must have heard these terms. You are all what? 21, 22, less than 25. Everyone is less than 25 years old here. So this is your
It means after. Ma'am, after. Sir, after. It means after or beyond.
meta means. Let me give you another example. How many of you use social media platforms? Instagram, Facebook. There also you must have heard the word meta, or those who are interested in computers must have heard the word metadata. Meta means on a very, very large scale, on a global scale, something which is huge, like which includes everything. Okay, so meta means like in totality or a lot or something entire. So entirety. Okay. And the word morph, always remember, the word morph means appearance. It's associated to appearance. Okay. So largely appearance. So if an organism goes through change in appearance on a large scale, so if you look at a caterpillar, so if I put a caterpillar at one end and tell you that it will turn into butterfly, something very colorful and beautiful, and the caterpillar is dull and worm-like, you can, even if you don't know the process, you can imagine that something must have happened very, very, like, on a big scale, like something changed completely. It transformed, correct? It's not easy to imagine a worm becoming a butterfly, but in biology it happens, and it happens on a large scale with respect to its anatomy and morphology. So it's called metamorphosis. So completely changing your appearance. Is it clear, everyone?
Yes, sir.
Yes, sir.
Yes, sir.
This term to your terminology section. We will be using this term a lot, and
Sir,
just because, yes, Samson.
what is the difference between morphing and cloning?
What is the difference between
Morphing and cloning.
morphine? You're saying?
No, sir, morphing. You use the term morph here. So, what is the meaning of morphing?
Can you be a little more loud, or you can write it, because I'm not able to understand. I understood the next word was clone, right? There, Samson, you just have to be a little louder. Can everyone hear? he has written. Morphing and cloning. Okay, beautiful, good terms. So Samson is asking what is the difference between these two terms. Sorry. Morphing and cloning. We will be coming to cloning in this chapter only. So on a biological level, Samson, let me tell you, there is a lot of difference in the process, in the how part, than the what part. So can I just keep this question for a little longer? We'll discuss this question when we'll come to cloning. Okay, will it be okay for you, Samson?
Yes, sir. Yes, sir.
Yeah, good question. We'll come to morph. So let's first talk about morphosis. So wherever in biology you hear the word morph, morphogen, morphosis, or morphology, you know that it is related to appearance of some or the other kind. Is it clear, everyone?
Yes, sir.
Yes.
Yes, sir.
Metamorphosis is the process by which organisms like insects go from their larval form to the adult form, showing a total transformation in their appearance. Okay, so we use the word metamorphosis. We also use the word molting for the same process. Molting specifically for insects. Okay, metamorphosis, molting. There was one more word for this kind of a development. So this is about indirect development. Is the concept clear to you? I'm not defining it here, just making a foundation. I'll define it when it will be required. But everyone gets the concept of indirect development?
Yes, sir.
Yes, sir.
And why the definition has adult in it? Because offsprings have to be adult so that they can be compared. The other one is direct development. Okay. Now direct development happens in most of the organisms that give birth. When they give birth to the new young one, that organism is already developed to the parent's form. Okay. So direct development happens in humans, in all the mammals, even in including your cats, dogs. So their babies look like their babies. You can say that. So direct and indirect development is clear, right, everyone?
Yes, sir.
Yes, sir.
Before going forward, I also would like to discuss with you all few things that will help us along the way. One is, how many of you know about DNA? What is a DNA? What is a gene? How many of you know DNA and gene? These terms, are you clear with this? DNA, gene, chromosomes. Again, terms that are difficult to remember, but once you understand it, it's easy. Anyone? DNA. What do you understand by DNA?
So it's like a self-replicating process that
Cell.
happens from parents to their offspring.
Self-replicated process. Okay, but what is it? I'm asking the question, what here?
It is a cell that contains genetic information.
Yes, it is not the cell, it is inside the cell, Shin. Okay. So DNA is present in each and every cell of every organism's body, even if it is a single-celled bacteria, it has DNA inside it. Okay. And inside the cell also there is a specific compartment in eukaryotes, class 11, remember? Eukaryotes have a true nucleus, which is membrane bound. The nuclear membrane is there. And inside the nucleus sits the DNA. We call it the blueprint of life, something that biologists like to experiment with a lot. If you go into research, DNA is going to be your favorite word. But even if you become a doctor and go into therapy, still gene, genetics, and DNA has to be dealt with. There is a reason that we cannot put anyone's blood in anyone's body. And there is a genetic reason for it. And the reason lies in DNA. Anyone else? DNA? So two things came from your side. Shin said that it's, what do you say, Shin? It's important for what?
Genetic information.
Genetic information. Perfect. Right. But still the question is, what is DNA? Is it something physical or is it just a concept, imaginary thing? Like in maths, we always assume let X be this. Is it something which we are doing in biology, or is it something actually present? Yes, Fatima.
It is actually present.
Yes. What is it then?
It's a protein compound.
It's protein. Who else agrees with this thing? Is it a protein? So protein is one kind of a biomolecule. Again, biomolecules, we have studied the chapter, but if you feel anything that you are struggling with as concepts, we will take care of it in this class. Okay. We will uncover the syllabus to cover it. I'm not just going to put a terpene on the syllabus and say, okay, it's done. By the end of these classes, you have to be a biologist. Yes, so it's not protein, okay? DNA itself is another biomolecule, and DNA stands for, anyone knows the full form? If you know the full form, you know what it is. The what part gets?
Deoxyribonucleic acid.
Yes, sir. Correct. Deoxyribonucleic acid. Correct? DNA. So one thing, let's go to the terminology again and put DNA in your head forever. The term itself tells about it. The what part gets clear if you understand the term. See, it has, the first thing says deoxy. Deoxy means deoxy. You have read this term a lot in chemistry. Deoxygenation reaction, remember?
Yes, sir.
Yes. So the word D means deforestation. What does the word deforestation means? Quick.
Cutting off trees.
Removing forest, right? Deforestation. You cut the trees, there is no forest anymore. Correct? De-evaluation. You are demotion, promotion, demotion. The word D means always taking out or removing or degradation, correct? So deoxy means something which do not have oxygen, because that oxygen is taken away. Deoxy. Clear? Now what does not have oxygen? Ribo. So this deoxy word comes from, it lacks one oxygen. It lacks oxygen. The word ribo comes from ribose sugar. DNA is made up of two things. One is ribose sugar is there, and ribose sugar, just like any other form of carbohydrate, glucose, fructose, lactose, galactose, ribose, they are all sugar. Whatever ends with O's is mostly sugar in biomolecules. Is it clear, everyone?
Yes.
And it's a 5-carbon sugar. See, this is the foundation, important for boards, not for your, important for entrances, not for immediate boards, but let's discuss this. It's a 5-carbon sugar, okay? So you just have to know it, 5-carbon chain. Last word is nucleic acid. It just tells about the nature of this molecule. It is a acidic molecule because it's nucleic acid. You all must have read in class 11th functional groups in chemistry, type of substances, aldehydes, ketones, carbohydrates, remember, carboxylic acids, correct? So this is nucleic acid, which means DNA as a molecule in nature is acidic, correct? And if something is acidic, tell me what is the charge on that molecule? All acidic molecules are
Negative.
negatively charged. And you can say why, just giving an example. You all have studied this group in chemistry, COOH. You must have been studying this COH. Acetic acid, CH3COOH. Yes or no?
Yes, sir.
Yes. Someone should just keep saying yes so that I know that you are understanding. So if you take this out, it will be, there will be negative charge because of this oxygen, correct? So nucleic acids, DNA, because of a lot of proteins also present on it, is negatively charged as a molecule. So just remember that it's an acidic molecule and it is negatively charged in nature. These two things are going to be useful in upcoming discussions. Now, what is a gene? Quick.
Gene is the genetic material.
Gene is the genetic material or DNA is the genetic material? I think DNA was the genetic material till now.
DNA
Sir,
contains
DNA.
genetic material, right?
Yes, Fatima, let you finish. Then
DNA contains the genetic material.
DNA contains the genetic material. So what is the genetic material?
The gene.
Okay, Arpit.
Sir, I was saying the same thing. That it
You saying the same thing. Okay. So a lot of biology is about imagination. If you have good imagination power, biology will become easy for you. So imagine a thread, okay, a thread which we use to stitch the clothes. DNA is like a thread. It's a very long molecule. It's a biomolecule which is a polymer. It's a long biomolecule which has small linkers. Just like in a chain, you have linkers that attach together to form a long chain. Imagine a molecule like that, which is DNA. Okay. So where do you think gene is? Is gene something which is present outside DNA? Shane, yes.
Is present in the chromosome, right?
Gene is present on the chromosome. Where is DNA present?
Gene is made up of DNA.
Yeah.
Gene is made up of DNA. Okay. Many good answers coming. Gene is present on chromosome and gene is made up of DNA. Correct? Is
Sir,
any
समझ नहीं आ रहा.
no. That is what we are trying to do. I'm confusing.
that's becoming confusing.
Yes, that's deliberate. There were two statements given by you. Gene is present on chromosome and gene is made up of DNA. Okay, out of which the first very correct statement is yes, gene is DNA itself. It's just the fragment, a part of DNA. Is it clear, everyone? If imagine, I don't have something to show you with me right here, but let's imagine this pen is one DNA. Okay, imagine it as a long thread which is going in both the directions. Is it clear?
Sir.
Or let me use this pen. So this pen is a long thread of DNA, and this cap of it, which is actually the DNA itself, it is a part of DNA, the molecule. But if I just take this section out of the DNA, this could be one gene. Is it clear, everyone?
Yes, sir.
Yes, sir.
Completely clear to everyone?
Yes, sir.
So gene is basically a fragment of DNA. Now the question comes, how do we decide, like, is everywhere on the DNA are there genes? So the question to that is no. Genes, functional genes, constitute only less than 1% of the total DNA length that we have in our body. Now this is ridiculous to hear in the first go. Many of my students have just heard this and said, what? Why? Because you must be feeling that if less than 1% of the total DNA length is gene, then what is the rest 99% doing, correct? Right?
Yes, sir.
Yes, sir.
Yeah. So it has a lot of regulatory and modulatory functions that we still are finding out. Like it's in the process of being discovered. A lot of DNA, the whole DNA is yet not understood completely. But just remember that if this is a DNA thread going, then from this part onwards to this part onwards, we call it gene A. This is a gene responsible for, let's say, eye color. Okay. Now, how do we decide that where the gene starts and where the gene ends on a DNA? Now, for this, we have an answer. Every gene starts with it has an open reading frame. We call it ORF. Open reading frame. Very important from your genetics point of view. Now, this open reading frame or ORF has, in the starting, there is a start codon. Now, I am assuming for now that you know what a codon means. You all know that DNA has three things. DNA has three components. One is the sugar, ribose sugar. The other is a phosphate group. Just imagine it, phosphate group. These two together form the backbone. Just hear it, I'll explain it to you. I'll give analogies, don't worry. And then there is a third component, which is called nitrogenous bases, or simply bases. These three things together form a nucleotide, which is the one linker. Remember I was telling you about, so if you have seen chain looks something like this, correct? It has many linkers that link together and form a long chain, correct? Everyone?
Yes.
So this one unit, this one linker of a DNA is this nucleotide. So for entrance point of view, there are three things. Remember, nucleotide is a single unit of the long DNA polymer. Nucleotide is made up of three things: a sugar, which is ribose in case of DNA, a phosphate group, and a nitrogenous base. And they are arranged something like this. So there is a backbone, and this you all know that DNA has a double helical structure, right? Double helix, the
Yes,
word
sir.
double helix, you all have read, correct?
Yes, sir.
Now it is arranged something like this. So how does a double helix look like? A double helix looks something like this. So this is one.
Twisted ladder.
Sorry,
Sir, it looks like a twisted ladder.
a twisted mat.
Sir, ladder, sir.
Ladder, yes, correct, perfect. So this is how a double helix look like, a twisted ladder. This is what you were telling, right? If you see books, you will see DNA represented like this, correct?
Yes, sir.
Just pay attention that I'm making two things in between in pairs. So these two things which are running like this are forming the backbone, sorry, this. So these are two backbones, and from these backbones will come the steps of ladder. Is it clear, everyone?
Yes.
Now these backbones are made up of ribose, sugar, and phosphate group. Clear? So there is
Yes.
one sugar, one phosphate, one sugar, one phosphate, one sugar, one phosphate. And a series goes like this: sugar, phosphate, sugar, phosphate, sugar, phosphate. And from every sugar towards the inside goes a base, which forms the step of the ladder. Is it clear to all of you? All of you can see me, right?
Yes, sir.
Yes.
Yes, sir.
So this is the backbone part. This here is the backbone part. And this inside is where nitrogenous bases are present. So one base from one side and other base from other side comes and pairs up in the center with hydrogen bonding. Now you all know there are four types of bases. If you don't know, it's fine. They are called A, T, G, and C. Remember? Anyone remember hearing this? A is for adenosine, thymine, guanine, sorry, adenosine, thymine, guanine, and cytosine. So these are the four bases, and we represent them by their first letter A, T, G, C. So if I take this portion of DNA and write it, like express it for you, we will read just the bases: 1, 2, 3, 4, 5, 6, 7, 8 bases, and we can write A, T, G, C, E, C, G in any sequence. So this is called the sequence of DNA. Is it clear, everyone? So DNA is a molecule, again a very long polymer. Each unit is called a nucleotide. The nucleotide is made up of sugar, phosphate, and nitrogen. You don't have to mug it up. I can promise that after 1 year it will be just in your head. And there are four different types of bases possible at every position. Either it can be A, T, G, or C. But it's the long sequence that has millions of these bases, and when we write the DNA sequence, we just write these bases. Is it clear? Because everything else is same. Makes sense to you, all of you?
Yes, sir.
Yes, sir.
Perfect. Now this is where biologists come in and tweak with the DNA. We read DNA as this. Now DNA is always read in the pair of, like not pair, in triplets. So this 3, then this 3, and this another 3. There will be one more. So this 3. So it's always read in triplets. Okay. And one particular triplet is called a codon. Is it clear till here, the story? Say yes or no?
Excuse me, sir.
Yes, sir.
Sir, is this sequence fixed? कि अब इसी में हमको read करना है?
Yes, so for genes, it is conserved. In humans, see, there is slight variation in every human with respect to every human, but that variation comes in the non-functional part mostly. Remember I told you 98% is regulatory part? How
Yes.
that plays a role defines how are we tolerant towards diseases, what is our immunity, which genes are active, which genes are not active in our body, which kind of proteins are made, whether we have lactose intolerance, allergy to dust, everything about us is shaped by the DNA. Is it clear?
Okay, sir.
But sir, the sequence doesn't have to be same, right? Like ATGC, ATGC, it's not supposed to be like that.
No, you mean to say ATGC, ATGC, it could be anything. It could be any combination of ATGC.
Okay.
If I give all of you a task to make a 8 alphabet long sequence of DNA and make all the permutation combinations possible, there'll be a lot of sequences, right?
Yeah.
Yes. So that's true, and that's what make DNA such a versatile molecule, because if you fix the reading, then the DNA will get limited. Then there will not be many genes. So we are talking about genes. For that, I wanted you to understand DNA. For that, I wanted you to understand nucleotide and the codon. So it's always read in 3 letters, 3 alphabets, and it's called one codon. Is it clear?
Yes, sir.
Yes.
Now coming back to a gene is defined as every gene has an open reading frame, and every open reading frame, it's called open reading frame because it's open to read and know and make changes. Like biotechnologists do that, you know. CRISPR gene editing techniques. CRISPR is one thing that's in hype a lot. Recently, the scientist who made a gene edited baby got released from prison in China. So what he did, without the consent of the scientific community, he designed a baby. And by designing, I mean he tweaked certain genes, because now there are techniques where you can, because we know the gene, we know the open reading frame, we can make biological scissor molecules which will go cut this, because this is all chemistry, correct? These are all molecules, so lots of chemistry is happening. So we can send something which can cut the bonds, take that codon or that gene sequence and put something else in between. You understand? Fancy, no? But that's the future.
Yes, sir.
So this technique is being developed for therapies, for getting rid of many diseases. So one day in near future, people will not have diabetes at all, because the gene that is responsible, you can just get rid of it, or you can put a new gene in. That's called gene therapy.
So until now, why it hasn't been discovered? Means like at what stage are we?
It's been discovered and we are at a very advanced stage. We are doing experiments, like my MSc research was based on CRISPR, but I was doing it on embryos of fish. There's fish called zebrafish. So we were editing genomes of fish to look for a gene of our interest, what it does. So in biology, there are 2 ways to find out what this thing do. Can anyone tell me? And it's logical. If I ask you, what is the importance and what is the role of Arpit in this group? How will we figure that out? Anyone? If I ask you that you have a group of, let's say, 5-10 people, what is the significance of Arpit in this group? Then how can I assess that significance? How can I figure that out? What will you do as the team leader? Simple. What will you do? It's simple. When do we get to know the importance of mothers or fathers or anyone in our house, or cleaners?
After we lose them.
When they are not there?
Exactly, when they're absent. So how are we doing in their absence tells us what was their significance, correct? So if there's a new gene discovered, you don't know what that gene does, you just knock that gene out of the organism. And if that organism just cannot survive, you know that that gene is vital for survival. If the organism survives but is blind, you know that this gene was playing some role in the formation of eye or something like that, correct?
Yeah.
So that's called knockout procedure. So either you knock out a gene and see what is happening, or you knock in something somewhere else, right? If I want to know what Arpit can do in a group, I can just put Arpit from this group and put it in some other group and see what he's like what his contribution is in that group, correct? So either you can take something out and see what's missing now, or you can put something in and see what's being added up. Correct?
But this is not like allowed now, because it is going to give harm to the organism.
Yes, that's not allowed for humans and for like animals. Largely, we cannot just do it for fun. As scientists, we have responsibilities, and for every organism that we are sacrificing for science, there have to be proper procedure. There have to be licenses. There have to be, if you are using a mouse for an experiment, you need to get a license from the required agency or the authority that I will be using a mouse. And after every experiment, whatever experiment we do on organisms, we do not leave them in the wild. Can anyone tell me why? Again, biology, basics 101. Why don't we leave? We just take organism, do the experiment, get the results. Yes, Fatima.
Because the gene is changed and many changes can come in the organism.
Yes, the organism can go on and pollute the already existing gene pool in the nature. Because we tweaked with its gene, we do not want that tweaking to happen everywhere in the wild, right?
Yeah.
Because that will be catastrophe in nature on a bigger scale. So a lot of regulations have to be there in science. But this person, being a scientist, just did one thing. He edited certain genes of a baby, of an embryo of a baby, which was then transplanted through IVF in a female, and she gave birth to the babies, where he later declared that I tweaked certain genes for obesity. So now these babies will never be obese. But who asked you to do that? You know, that's not ethical.
Isn't that good?
Yeah, he will say it's good. Like, of course, the baby will never be obese, but we do not yet have reached a level where we can with surety say that the baby will only not be obese without any side effects. You understand?
Ooh, yeah.
For any research to happen and become translatory in biology, we have to do it multiple times on different model systems and be sure that it will work. Then it reaches humans.
Excuse me, sir.
Yes, Shen.
I have to go open my fast, so can I leave?
Yes. I'll be just I'll be ending the class in 10 minutes, so you will not miss much. You can go. Okay?
Thank you, sir.
So anyone else who needs to just leave, or you can stay for another 10 minutes, I'll just finish in 5-10 minutes. So every gene, we were coming back to genes. So you understood, Fatima, why it's bad, why he was jailed, and he was jailed for a significant term, like around 7-8 years he was in jail. Because you cannot do this without proper consent and ethics. So every gene starts with a start codon, which is always AUG. I remember it for August. AUG is for August, and my way to remember it, because in August India got independence. It helps me because of my nationality. So India started from August, so every gene starts from August. Cool?
Yeah.
Easy to remember. So AUG is the start codon. So every gene, wherever you have to find where are all the genes, you have to do two things. Look for start codons and look for stop codons. So the second one is a stop codon. Every gene starts with a start codon and stops with a stop codon.
What is the stop codon?
Yes, there are 3 stop codons. Either it could be UAA. Now U is, you did not ask that, I did not tell you about U. I only say ATGC. Where does U comes from? Right? No one asked that. Okay. Let me ask myself. T stands for thymine, okay, in DNA. So in DNA, there is thymine. In RNA, there is, in place of thymine, there is uracil. Remember that T is not present in RNA, it's present in, only in DNA, right? And whenever we read a gene, we only read the half only one strand. Look here. So there are two strands. If I just break it in the middle, there will be two strands, right? That's why it's called double helix, correct?
Okay.
So DNA is double stranded, RNA is single stranded. From DNA we make RNA, from RNA we make proteins, and proteins are the functional unit in every cell.
Sir,
Yes, Samson.
what is RNA?
Yes, good question. Thanks. DNA is the genetic material. Okay, everyone?
Excuse me, sir.
Yes, Arpit.
Sir, the starting codon is always same. It's
Starting point of gene is always same. Yes, for every gene it has to start with AUG. That is the start codon. But everything in between will be different, right?
But can we change the start codon?
If you change the start codon, it will not be start codon then. I will tell you why it is called start codon, but I will teach you in detail in biochemistry, the part that we will cover later in 12th. So think of start codon as iCards. So for every institute, there is only one kind of iCard, right? The type, like now the details, whatever is there in the name, blood group, father's name, address, your course is different. But the format of that iCard, which you can use to enter, is same, correct?
Yeah.
yes, sir.
If I allow everyone to make their own iCards, whoever is fancy makes fancy iCards, someone will just write on a piece of paper, this is my iCard, and bring to the college, then it will not work. So to start any gene, and why these genes are there, because ultimately from a DNA, which is a gene, we have to make RNAs. RNA is ribonucleic acid. The deoxy part is not there. This is deoxyribonucleic acid. This is ribonucleic acid, RNA. Is it clear, everyone?
Yes, sir.
Yes, sir.
It has oxygen. So from DNA, we make RNA, and RNA is single stranded. It has just one strand because that is enough to read the gene. And from that RNA, we make proteins in the cell. Okay. Now this part is important. Pay attention here because this is called central dogma, very important for entrances. See, DNA can replicate and make more DNA. Say yes.
Yes.
Yes,
This
sir.
process of self-replication is called replication, DNA replication. Someone in the beginning used this term, DNA is a self-replicating system. Who said that?
Me, sir, I said that.
Yes, Samson said that. So this is where this part comes. DNA can replicate and make another DNA. From the DNA, we can make RNA. Now, RNA cannot make another RNA normally. This cannot self-replicate. It can only be made from DNA. And from RNA, we make proteins. So this part is called replication. From DNA to RNA is called transcription. Look at the terminologies again. Let's come to that. Replication is a simple English word. Replicate something, right? Make a copy, replica. Transcript. What is a transcript? Transcription.
DNA to RNA.
Transcription in normal language means something was written in Arabic. We converted that same text into English, the same information. We transcribed it, right?
Yeah.
So the same information which was in DNA is made into RNA, transcribed from one language to another, so that some molecules in the cell, some important molecules which are called the protein synthesis units in the cell. Now, who will tell me which are those organelles which produce protein in the cell? Protein factories.
Mitochondria.
Mitochondria is a powerhouse. Protein synthesizing units starts with the same alphabet with which my name starts.
Ribosome.
Yes, perfect. Ribosomes read RNA and make proteins out of it. Remember, class 10th is getting connected here in DNA.
Yes, sir.
Biology is all related. So we need to make the RNA from DNA so that ribosomes can read this RNA. Transcription is made and then translate it into protein. So that's called translation. Replication, transcription, translation. Is it clear? And this is what every living system follows. This is called central dogma of biology, the central rule of biology. Is it clear, everyone? The basics, foundations?
Yes, sir.
Now you should know that there is one system on this planet that can make DNA from RNA. This is possible under very, very limited special conditions. And there is one, I should say, organism or a system that can do it. And that system is called a virus. Virus can make DNA from RNA. So if making RNA from DNA is called transcription, what will be this process called? Simple logic. If there is swing in cricket, then there is also a reverse swing, right? Say yes or no. Gear and reverse gear, osmosis and reverse osmosis. All the
Yes, sir.
purifiers in your home have reverse osmosis. RO system stands for reverse osmosis, correct? So if this is called transcription, what will this process call?
Transcription.
Reverse transcription. Okay, so you can write it. Reverse transcription is done by viruses. Is it clear?
Yes, sir.
We'll also come to viruses later. Okay, so gene is clear. Gene is a part of DNA. It starts with a start codon and ends with stop codon. There are three types of stop codon. Sorry, one was UAA, another was UAG, and UGA. Now, for this, it's a funny sound. It's UA, UAG, UGA. UGA, UAG. So U and A has to be there, and UA and G has to be there. Clear to everyone?
Yes, sir.
Yes, sir.
Okay, very good. So some basics of DNA and gene is clear. Now you already imagined. I will finish with chromosome and we'll take from here and we'll begin with reproduction in the next class. So you imagine that DNA is a long thread, correct? And gene are different segments of DNA only. So gene is also basically part of DNA. How many of you in your homes have seen how do we keep threads? How do our grandmothers or mothers used to keep wool?
In bundles.
In
Bundles.
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 What kind of bundles? They are called condensed. Like we just roll it over something or
Yeah.
roll it over itself and keep it into a small space. So a very long thread of wool or a long thread can be kept in a very small area. That's called spool, right? Spooling of wool or spooling of the thread.
Yes.
That is chromosome. So DNA is very long, very, very long. In fact, if you take all the DNA from all the cells of a single human and just align it end by end, it will reach the extent of our solar system. It's very, very huge. But DNA is also very thin.
And one DNA is how long?
In one cell, the DNA coming from both the parents, for a 2n cell, it is 1.3 meters in one human diploid cell. But the cell itself is micrometers in diameter, right? So it's like, I ask you, you have to keep 1 kilometer long cotton thread in a matchbox. So how will you do that?
We'll roll it. Yeah.
Yes, you will condense it as much as you can so that 1 kilometer long thread can just get condensed and fix in a matchbox, correct? This is what happens in cell. So that condensed form of DNA is called chromosome. Is it clear, everyone?
Yes, sir.
Yes, sir.
Please keep writing certain things, because when I'm discussing, I may not dictate or write everything for you, but you have to keep writing. So, and as we all know, to make spool of thread, we also need something on which we can roll it, right? In a chromosome, that part is played by certain proteins. So chromosome is not all DNA, it is DNA and protein together, because that protein helps in condensing the DNA. Is it clear to all of you?
Yes, sir.
Yes, very good. So let me just finish the class here because it's time for this batch. So please go back. I'm giving you one assignment before you come back yesterday. Yesterday we'll be taking from here, but you have to brush up in order to understand reproduction well. Two things. One is meiosis. I'm sure you all have read meiosis and mitosis in class 11.
Yes, sir.
So go and revise meiosis and mitosis. Gene, chromosome, and DNA is already clear. Revise this as well. And also from class 11th, revise alleles. Mendel. In the Mendel chapter, there was a term called allele. Okay. In the next class, I'll be taking you to different modes of reproduction, and we'll start with what actually NCERT wants us to understand. Okay. Anyone has any doubts? I hope you like the class and I hope you learned something.
Sir, you didn't give answer to my doubt.
Yes, we'll come to your doubt in tomorrow's class, cloning and the morphing part, right? So I want to cover cloning in as a part of asexual reproduction. Okay. So there, when I will be covering cloning, I'll also cover morphing for you. Will it work for you, Samson?
Okay, sir.
Okay.
Sir, the first chapter which we are doing is human reproduction, right?
We are doing reproduction. You want me to go first with sexual mode or asexual mode? I can go with any, depending on what students need for their schools. If you need human reproduction first, I can go with that. But normally I start
The first, flower reproduction and flowering plants. First.
That is also sexual reproduction, but that's just in plants. So when sexual and asexual. So let's start with asexual because that is the basis and the foundation for sexual. Okay.
Yes, sir.
That way we can also discuss a lot of things required for entrances.
So
Sir?
Yes, Fatih.
this asexual reproduction, does it come in any of the chapters?
Yes, it's the first chapter. First chapter is reproduction. It has two parts, asexual and sexual. In asexual, it talks about your vegetative propagation and budding, fission, all of these things. In sexual part, it talks about flowering plants and human reproduction. Clear?
But that part is deleted, right?
Is it deleted from which part is deleted?
The first two chapters.
Asexual reproduction is deleted from your syllabus? All of yours?
No, but
First chapter.
no, CBSE didn't give any information.
See, even if it is
Yes,
deleted,
sir, not
course
yet,
has
sir.
to be-
Sir, it is not deleted yet. There's no instructions from CBS.
even if it is deleted from your boards, believe me, it's going to come in your entrance, correct?
Yeah, yes.
We will prepare from both points. So let's just go this way and we'll see, okay?
Okay.
Sir,
Thank you, sir.
sir,
Yes, Arpit.
sir, the next class will be on when it will be.
Tomorrow, right? Saturday, Sunday. This is the weekend batch.
Okay, sir.
Yes, so today and tomorrow at the same time. See you tomorrow, everyone. Go back and just revise everything I have told you and also revise meiosis, mitosis, and alleles with genes. See you all tomorrow.
Thank you,
Thank
sir.
you, sir.
Bye.
Thank you, sir.
Take care. Bye.
Yes, sir. Bye.
Okay. Huzaifa, Unib, Yashika, and Zaid. Can you hear me?
Yes, sir.
Are you here for biology class?
Yeah, I am here for biology.
Yeah, Zaid is from the batch. So Zaid, yours one will start at 6:30, right?
Yes, sir.
Right now, like mine.
Right now.
Huzaifa, Unib, Yashika, are you also there for biology class?
No, sir, we're from math.
Okay, so yes, so Zed, so I'll finish. I'll log out from this class. The maths teacher will join, and Zed, you need to log in through the other.
Okay, sir.
Yeah. Okay. Thank you.
Sir, I think now I am having maths